Hello everyone. Today we are diving into the basics of lipolysis and fat oxidations. These processes are critical for understanding how the body breaks down and uses fat for energy, particularly during exercise or fasting. Let's start by defining what these terms mean and how they relate to our metabolism. Lipolysis is the first step in this process. It's the breakdown of stored fat in the form of triglycerides into free fatty acids or FFAS and glycerol. This occurs in adipocytes, which are fat cells, and is initiated when the body needs energy. Think of triglycerides as the stored form of fats, and lipolysis is how the body taps into this energy reserve. Once lipolysis releases free fatty acids, these fatty acids are transported to the mitochondria within cells, where fat oxidations takes place. Fat oxidations or beta oxidations is how the body turns and burns these fats to generate energy in the form of ATP or adenosine triphosphate. Essentially, lipolysis is the breakdown and fat oxidations is where the actual energy production happens. Understanding the roots of these scientific terms can make them easier to remember. For example, adipocytes come from adipo, meaning fats, and cytes, meaning cells. Triglycerides have a prefix tri, meaning tree, and glycerol refer to the sugar-alcohol backbones that connects the three fatty acids. As for glycerol, it's come from the root glycerol, meaning sweet, due to its discovery in sugar-base reaction, and O refers to its alcohol group. Glycerol is a sugar-like alcohol, which makes it key in fat metabolism because it resembles glucose in its structure. Now, let's talk about beta oxidations. This process occurs at the second carbon atom of a fatty acid chain, which is why it's called beta. Through beta oxidations, fatty acids are burned to produce ATP, the energy currency of the body. As for ATP, it's what's power our cells. It consists of adenine, a nitrogenous base, combined with ribose, a sugar, and three phosphate group. When one phosphate group is broken off, energy is released. Now, let's take a quick look at adenosines. This word is made of aden, which comes from the Greek word gland, because adenines, a component of adenosines, was first discovered in glandular tissues. The suffix "-ocines" refers to a sugar-bound nitrogenous base, indicating that adenosines is a combination of adenine and ribose, a sugar. So you can think of adenosines as a unit formed from adenine and sugar, and it serves as the core structure for ATP, the energy currency of our cell. A quick note on the history of adenine, which play a key role in ATP. Adenine was first discovered in gland tissues in the late 18,000s, and its role in energy production to ATP was established in 1929. By 1953, Watson and Crick confirmed adenine's roles in DNA as it paired with thymine. Today, we understand adenine's essential role not just in genetics, but also in cellular energy and signaling. Moving on to the history of fat metabolisms. In the 1890s, Claude Bernard explored the role of fat as an energy source. By the 1960s, the enzyme hormone-sensitive lipase was identified as key to breaking down triglycerides. In the 1980s, fat oxidations became a critical area of studies in exercise physiology, and today we continue to explore the genetic and hormonal factors that influence metabolism, particularly in relation to obesity. Now we're going to move on to the hormones and how these hormones play a crucial role in regulating lipolysis. Let's quickly look at the four key hormones. Epinephrine promotes lipolysis during fight or flight response. Insulin inhibits or stops lipolysis and promotes fat storage, especially after a meal. Glucagon stimulates fat breakdown when blood sugar is low. And cortisol has a dual role. It promotes fat breakdown during the short-term stress, but can lead to fat storage if stress persists. These hormones ensure that fat is broken down or stored according to the body's energy needs. Now, let's look at the enzymes. Several enzymes are involved in both lipolysis and fat oxidation. Let's look at the most important ones. Starting with the first one, hormonal sensitive lipase or HSL break down triglycerides into fatty acids and glycerols. The second is ATGL or adipose triglyceride lipase. It initiates the breakdown of triglycerides. The third is LPL or lipoprotein lipase, and it promotes fat storage by breaking down triglycerides into the bloodstream. Next is CPT1, is carnitine palmitoyltransferase 1, which transports fatty acids into mitochondria for oxidation. And the last is acyl A dehydrogenase is catalyze the first step of beta oxidation inside the mitochondria. Each of these enzymes play a critical role in either mobilizing stored fat or burning it for energy. As we wrap up, these are my notes for the next time that I'm going to study. So I'm going to talk first about the ketogenesis. This is where where fat oxidation exceeds carbohydrate availability and ketones are produced as alternative energy source. This usually happens when we fast. And exercise and fat oxidation. So yeah, I will have to explore about how fat oxidide became the primary source of energy when we are having like low to moderate intensity exercise. So there are also five common mistakes that I would have to look into, and as well as the three unusual case when it comes to lipolysis and fat oxidations. Thanks for listening. I hope this breakdown has made this concept clearer and easier to understand. Have a great day.